Welcome to Inert Chemistry. In today's video lecture, we are discussing about the biosynthesis of purine nucleotide. First of all, we will see what are purines. Purines are the nitrogenous bases and these are double ring nitrogenous bases. Here you see the general structure of a purine. These are 9-membered heterocyclic rings. There are two major examples of purine that are adenine and guanine. Adenine is also called 6-aminopurine because there is amino group at the 6th position, while guanine is also called 2-amino 6-oxypurine. Here is the structure of purine ring, and you see that a purine ring is actually a double ring and consists of 9 different atoms present in it. All these different atoms or elements, they are coming from different sources. For example, the nitrogen 1, it comes from aspartate. Aspartate is an amino acid and the amino group of aspartate donates this nitrogen. Similarly, carbon 2 and carbon 8, both these carbon, they comes from the folate of tetrahydrofolic acid. Nitrogen 9 and nitrogen 3 and nitrogen 9, they come from the glutamine. Carbon 4, 5 and 7, they are contributed by glycine. Glycine is an amino acid and these three elements, carbon, two carbons and nitrogen, they are contributed by glycine. While carbon 6 is directly come from respiratory carbon dioxide. Purine nucleotides. As there are two purine bases, adenine and guanine, and they form both ribonucleotides as well as deoxyribonucleotides. The ribonucleotides from adenine are AMP and deoxyribonucleotides are deoxy AMP or deoxyadenosine monophosphate. While from guanine, it is GMP, means guanosine monophosphate, and here is 2-deoxyguanosine monophosphate. Here we see the overview of metabolism of purine biosynthesis. There are three processes by which purine nucleotides are formed. These are de-novo synthesis, phosphoribosylation of purines, and phosphorylation of purine nucleosides. Out of these, the most important is de-novo synthesis, and here we discuss this. The starting material for the de-novo synthesis is ribose 5-phosphate. This molecule is formed by hexose monophosphate shunt, also called as HMP pathway. After about a series of 11 steps, it is converted into inosine monophosphate. This inosine monophosphate is shortly called as IMP. It is a parent nucleotide. The further different types of nucleotides like AMP, ADP, ATP, these are formed by different reactions from IMP. All these AMP, ADP, ATP, GMP, GDP and GTP, these are ribonucleotides. While deoxyribonucleotides that is present in DNA, they are formed from these ribonucleotides like GDP and ADP. As I have described earlier that purine biosynthesis consists of 11 steps. Let's discuss all these steps one by one in detail. This is the structure of ribose 5-phosphate. it is a 5-membered ring structure, and at the position 5 there is attachment of phosphate group. So it is called as ribose 5-phosphate. During the first step, an ATP molecule is utilized and it is converted to AMP, adenosine monophosphate. The two phosphate groups that are removed from the ATP, they get incorporated at the position 1 of ribose 5-phosphate, and the resulting compound is called 5-phosphoribosyl pyrophosphate, also called as PRPP. This reaction is catalyzed by enzyme PRPP synthetase. In the second step, this PRPP molecule is utilized, and here a glutamine is used. This glutamine donates the amino group to PRPP, and this pyrophosphate molecule is released. Here you see the pyrophosphate molecule is released and amino group is added into the PRPP. The resulting compound is thus called phosphoribosyl, because phosphate group is added at position 5, so it is phosphoribosyl, and due to the presence of NH2 it is called amine. Phosphoribosyl amine is formed. Here in the third step, this 5-phosphoribosyl amine is utilized, and then glycine, that is an amino acid, it comes into role. This glycine and ATP are utilized here. You know that glycine is the simplest amino acid, and it is incorporated into 5-phosphoribosyl amine. Here this glycine molecule C double bond O CH2 NH2, this is the glycine molecule. It comes and attach with NH by the help of enzyme synthetase, and the resulting molecule is called glycine amide. Because of addition of glycine, it is called glycine amide ribosyl 5-phosphate. In the fourth step, this glycine amide ribosyl 5-phosphate is used. Here methynyl tetrahydrofolate is used. This group is formyl group donor. This molecule methynyl tetrahydrofolate donates the formyl group. CH double bond O is a formyl group, and N5N10 methynyl tetrahydrofolate donates this formyl group to glycine amide ribosyl 5-phosphate, and the resulting compound that is called formyl glycine amide ribosyl 5-phosphate, because here is an addition of formyl group. In the fifth step, this formyl glycine amide ribosyl 5-phosphate, again glutamine comes into play, and here again an amino group is donated and it gets incorporated here. Here you see that here C double bond O, but here is C double bond NH. At this carbon an amino group is added, so it get converted to formyl glycine amide ribosyl 5-phosphate to formyl glycine amidine ribosyl 5-phosphate. Then this formyl glycine amide ribosyl 5-phosphate, it undergo ring closure reaction. Here an ATP molecule is utilized and this ring get closed. Here you see that this ring is closed. This ring that contains 2 nitrogen atoms, 3 carbon atoms with 2 double bonds. This ring is called as amidazole ring. This ring is called as imidazole ring. So this is your 5 amino imidazole ribosyl 5 phosphate that is formed here. In the step 7, respiratory carbon dioxide is added by the help of enzyme carboxylase, and it attaches at the ring amino imidazole ribosyl 5 phosphate रिंग में जो है वो कार्बन डाइऑक्साइड ऐड हो जाती है. देन इन द 8th स्टेप aspartate is added. This molecule of aspartate gets added into the amino imidazole carboxylate, and the resulting compound is called 5 amino imidazole succinyl carboxamide ribosyl 5 phosphate. Carboxamide is called because of addition of carbon dioxide, and succinyl due to the addition of aspartate. In the 9th step, the fumarate molecule is released from this molecule 5 amino imidazole succinyl carboxamide ribosyl 5 phosphate में से fumarate का molecule जो है वो निकाला जाता है. Here only NH2 remains, while the all other group is released in the form of fumarate. In the 10th step, the N10 formyl tetrahydrofolate donates the formyl group again. Here you see this C double bond OH is the formyl group. It is again added into this by the help of enzyme formyl transferase. And in the 11th step, again the ring closure occurs with the release of water molecule by the help of enzyme cyclohydrolase. This ring gets closed and this inosine monophosphate is formed. Here you see that this inosine monophosphate is formed on the molecule of 5 ribosyl 5 phosphate. This is ribose 5 phosphate molecule, and this nitrogenous ring or inosine monophosphate is formed on the molecule of ribose 5 phosphate. Here we have again summarized all these 11 steps for your convenience in one slide. Although it is very difficult to remember all these names and reactions in a sequence, that if you practice this, it will be very easy for you if you practice it for 2 to 3 times. Thanks for watching this video. In the next video, we will see the next part of this lecture, in which inosine monophosphate is converted to adenine monophosphate and guanine monophosphate.